Hello, welcome to World of Microbes. In today's video, we are going to discuss conventional diagnosis and molecular diagnosis. Considerable factors to decide suitable diagnostic methods, differentiate between conventional diagnosis and molecular diagnosis, conventional and molecular methods, and we'll discuss if molecular will fully replace the traditional microbiological diagnostics. Do like and subscribe. A fast but reliable identification of pathogens causing acute infections is a highly important issue in microbiological diagnostics. Conventional diagnosis refers to traditional methods of medical diagnosis, often relying on physical examination, clinical presentation, and basic laboratory tests. Conventional methods are well adapted and quite inexpensive but time-consuming and accompanied by certain limitations. While molecular diagnosis utilizes advanced techniques to analyze a patient's genetic material, DNA or RNA, to identify diseases with higher specificity and sensitivity, usually providing faster results and earlier detection of conditions, particularly at the molecular level. However, molecular diagnosis can be more expensive and require specialized equipment and expertise compared to conventional methods. Modern molecular methods have been increasingly incorporated in laboratories. Many research have been done on the development of rapid and effective diagnostic methods. New modern diagnostic test is widely accepted because of the quick and effective test results, within a short time duration, which will help physicians to correctly diagnose the disease for treatment purposes. However, cost of the test will increase because of expensive reagents and qualified staff to run the test. Some pathology labs still uses traditional methods because of cost effectiveness. Our main objective in this video is to compare conventional and modern diagnostic tests to understand the demand of the community for the successful acceptance of modern diagnostic methods. Here are some key differences. No. One is method. Conventional diagnosis often involves techniques like microscopy, culture-based methods, and serological tests, while molecular diagnosis uses techniques like polymerase chain reaction, PCR, DNA sequencing, and in situ hybridization to analyze acids directly. Next is accuracy. Molecular diagnosis generally offers higher accuracy due to its ability to target specific genetic sequences, leading to greater sensitivity and specificity compared to conventional methods. Next one is speed. Molecular diagnosis usually provides faster results than conventional methods, enabling quicker diagnosis and treatment initiation. Another one is application. Conventional diagnosis is often used for initial screening or when molecular testing is not readily available, while molecular diagnosis is particularly valuable for detecting infectious diseases, genetic disorders, and monitoring treatment response. Examples of conventional diagnostic methods include blood cell counts, urine culture, serological tests for antibodies, and tissue biopsy with microscopic examination. Examples of molecular diagnostic methods include PCR for detecting pathogens, next-generation sequencing for genetic analysis, in situ hybridization for detecting specific RNA or DNA sequences in tissue. Important considerations. Cost. Molecular diagnostics are often more expensive due to equipment and reagents required. Expertise. Performing molecular tests may require specialized training and expertise compared to conventional methods. Ethical concerns. Genetic information obtained through molecular testing raises ethical considerations regarding patient privacy and genetic counseling. Most considerable factors to decide suitable diagnostic methods are cost-effectiveness, specificity, sensitivity, availability of qualified staffs. Currently, nucleic acid manipulation and testing are common tests used in the scientific community. Promising approach involves advances in nanotechnology field actions to simplify assay procedures to make it user-friendly and enhance the detection sensitivity of the assay. Do like and subscribe. Here are the basic 4 steps of diagnosis. First one includes symptom diagnosis, in which signs and symptoms are diagnostic indications of an illness, injury, or condition. Signs are objective and externally observable. Symptoms are a person's reported subjective experiences. Next one is cultural diagnosis. It uses growing microorganisms in a laboratory to identify the cause of an infection. It's one of the main ways to diagnose infectious diseases. Immunity diagnosis, in which the standard screening tests for humoral immune function start with measuring immunoglobulin, Ig, or antibody, levels in the blood serum. These consist of IgA, IgM, and sometimes IgA levels. The results must be compared to age-matched controls. There are also tests for specific antibody production. Lastly, molecular diagnostics, a laboratory method that analyzes DNA, RNA, and proteins to identify diseases or the risk of developing them. It's also pathology. Do like and subscribe. These methods are highly sensitive, reliable, low-priced, and provide both qualitative and quantitative results on the bacterial populations present in the clinical sample. However, these samples are commonly mixed cultures, including diverse pathogens, but also microorganisms of the normal flora. Typically, the isolation of applying several growth steps is necessary to characterize the bacteria in more detail. Consequently, the conventional methods are labor-intensive and time-consuming, since the results are not available until at least 1 to 3 days. Moreover, the culture-based methods reach their limits with specific microorganisms that do not grow on or in artificial. Traditional diagnostics are based on the ability of the microorganisms to grow under artificial conditions. Some examples are microbiological culture using selective or differential medium, microscopy, gram staining, biochemical tests. Do like. Here are some examples of conventional methods of bacterial detection. Some of them are culture-dependent, whereas others are culture-independent. Do like and subscribe. Molecular methods substantially revolutionized microbiological diagnosis. These techniques are more convenient than conventional methods as they are generally faster, more specific, precise, and preceding cultivation of microorganisms is not necessary. In contrast to the conventional methods, the identification and culturable and slow-growing pathogens is easier to implement. In addition, they can be performed and results interpreted by staff with no taxonomical expertise. Moreover, they apply more stable genotypic characteristics for identification than traditional techniques using phenotypic characteristics. Examples of molecular methods: reaction, PCR, copies specific DNA sequences, which can be used to detect genetic mutations or pathogenic microorganisms. Real-time PCR quantifies specific markers in DNA samples, such as genes. Fluorescence in situ hybridization, FISH, detects and localizes specific nucleic acid cells. Ligase chain reaction, LCR, detects single base pair mutations. Molecular modeling, used to design drugs. Molecular typing identifies specific strains of microorganisms, such as bacteria or viruses. Uses of molecular methods. Cancer, monitors tumor burden, minimal residual molecular resistance. Infectious diseases, screens for bacterial pathogens, causing sepsis and respiratory viruses. Fetal medicine, analyzes cell-free DNA. Coagulation disorders, analyzes blood samples. Environmental diagnosis, identifies markers in DNA samples from the environment. Subject to regulatory standards that assess sensitivity, detection ability, and participation in proficiency testing programs. Do like and subscribe. Different molecular methods and techniques will be discussed in details in further parts of this video. Do like and subscribe.